Yeah, good evening, everyone. I'm Saranjeev Rangaraj. So I will start my presentation now. There was some issue with this recording option. Just now I resolved it. Yeah.
Okay.
So is it visible? My PPT?
Yeah, we can see it.
Yeah. Okay. Good afternoon. So I'm Saranjeev Rangaraj. I'm a PhD scholar from the Department of Biochemistry, University of Hyderabad, and also I'm a PMRF fellow to fulfill my teaching assistantships. However, I have been associated with this NPTEL team, and the course that we have been seeing now is the overview and then integration of cellular metabolism. And we are in week 11, and almost we have done with all the metabolism that we have seen so far. For example, lipid metabolism, carbohydrate metabolism, amino acid metabolism, and finally we have started with nucleic acids metabolism in the last week. So as usual, we'll see some recap on the week 11 concepts, and we'll do some practice question, and if you have any doubt, then we can go back and then see. But last week we have stopped with the how purine synthesized in the body, and then what are the regulation of the purine metabolism that we have seen. And this week we are going to start with the nucleotide degradation. And if you see, any nucleotide is composed of three compounds. One is a ribose sugar, and nitrogenous base, and finally the phosphate molecule. And any nucleoside, if it is in the presence of nucleosidase, it will go hydrolysis, and finally it will give the base as well as the ribose. And there is some more enzyme called nucleoside phosphorylase. What it does, it will change the nucleoside into base and then the ribose 1 phosphate. So we are going to see how these nucleotides are degraded in the body. One such example that if you see in any catabolism of purines, the respective AMPs, IMP, xanthan monophosphate or guanine monophosphate, in the presence of nucleotidase, that we know that in the presence of H2O, it will be converted to the respective adenosine. So here one molecule of this phosphate molecule will be removed, then the respective adenosine, inosine, xanthosine, and then the guanosine will be formed. And there is an enzyme here that is adenosine deaminase. What it does, it will change the adenosine into the inosine. And the same way, the xanthosine, in the presence of the purine nucleoside phosphorylase or the other phosphorylase, respectively, changes the inosine into the hypoxanthine and xanthosine into xanthine, and then the guanosine into the guanine. So these are the two enzymes that we have seen it in the last slide. And here the respective oxidase or then the deaminase will come that will change the guanine into the xanthine and then the hypoxanthine into the xanthine. So this xanthine is really, really important component in the body, where the xanthine oxidase, which contains the FAD and molybdenum and then the iron in the in its active center, then it will convert into the uric acid. So far we know that any purine compound, whether it can be AMP, IMP, or ZMP, or GMP, it will be finally metabolized or the catabolized to uric acid. And this is a one such example in the body where the direct reactive oxygen species is synthesized, such as H2O2, that is the hydrogen peroxide. And if you see the metabolic disorders related to this purine metabolism, it contains the hyperuricemia. Hyperuricemia is nothing but high levels of uric acid, and then the acute renal failure, and it also called as a gout, unexplained neurological defects, for example, seizures, muscle weakness, and etc. And developmental disorder takes place, and compulsive self-injury, aggression, and immune deficiency, and deafness takes place. The major disorders can be of two types in the purine nucleotide metabolism. One thing is it can be a blockage where the A substrate will not be converted to the B product, or as the increased activity of certain enzymes that leads to the high level of accumulation of the product. So the blockage of the degradation occurs with the symptoms involving the immune deficiency, myopathy, and then the renal calculi. And there are increased degradation of the nucleotides occurs where with the symptom characterized with the hyperuricemia and then the gout and then the renal calculi and then the anemia, acute hypoxia. So we have seen uric acid, that is the final product in the catabolism of purines. The uric acid is nothing but which is 2, 6, 8 trioxypurine, and it accumulates in the blood, that is referred to as hyperuricemia. So the reason why it is really difficult to treat, uric acid is a highly insoluble in the body, so it's very slight alteration in the production of the or solubility will increase level in the blood. The reason is the uric acid is very insoluble. Due to this poor solubility, what it will do, it usually sediment in the places where it should not be sedimented. Usually, the excretion of the uric acid takes place through the glomeruli, and more than the 80% of the uric acid formed in the urine is derived from the distal tubular secretion. But whereas the high level of uric acid synthesis in the body takes place, that leads to the sedimentation of this uric acid as a some monosodium salt that we are going to see it in the next slide. So the hyperuricemia is nothing but in any perfect health man, he has less than 7.0 mg per dl uric acid in the blood, whereas more than in women it is less than 6.0 mg per dl. Whenever it crosses the threshold, like that is more than 7.0 mg per dl in men or more than 6.0 mg per dl in women, that will be termed as hyperuricemia. This can occur in the two ways. One is the increased production of the uric acid or the decreased excretion. So the factors contributing to the hyperuricemia, it can be the synthesis of purines. The more purine synthesis leads to the more uric acid synthesis, and then the secondary gout, which will consider as the disorder related to other metabolism that leads to the rapid tissue background, and that is helps in the rapid cellular turnover. So the cellular turnover leads to the more purine degradation, and finally uric acid synthesis will takes place and increases the intake of purines and then the increase in the turnover of nucleic acid that we have already seen. And there are other factors which influence this hyperuricemia that can be said. When usually we know that male usually have more uric acid than the females, and then the obesity, the obese people tends to high plasma levels of urate, and the diet in the sense subject with the high protein diet or which also rich in the nucleic acids and who have high alcohol consumption have high levels of plasma uric acid. And genetic factors are very important. These are very important factors in the high plasma urate levels. And the conditions that leads to the increased uric acid production, if you see, if you have any kind of malignancies, Reyes syndrome, Down syndrome, sickle cell anemia, glycogen storage diseases, and hereditary fructose tolerance, and then the acyl CoA dehydrogenase deficiency also leads to increased uric acid production. The gout is nothing but it is characterized by the accumulation of the monosodium urate crystals deposit, which result in the inflammation in joints and surrounding tissues. And this presentation related with the hyperuricemia, uric acid nephrolithiasis, and then the acute inflammatory arthritis. So, if you see the picture says the accumulation of the monosodium urate crystals the different places, like it can be in the toe or it can be in the knee or it can be in the ear and it can be in the fingers. So this leads to the high level of pain because these are not insoluble in the body, and sometimes it due to the gravitational force it will be sedimented in the knees as well as in the toes of the body. There are two types of gout usually we see. One is the primary gout as well as the secondary gout. The primary gout is occurs usually in the middle-aged men mostly, and secondary gout usually occurs in the children and then the adults. The cause of the primary gout, if you see, the overproduction of the uric acid and then the decreased renal excretion or both. The biochemical etiology, if you see, that there is no clear cut evidence that this is happening because of the A reason or the B reason that you can state, because it is a polygenic disease. This is leads to the multiple reasons are related to the primary gout. But whereas this secondary gout is the condition where the rapid tissue breakdown or the cellular turnover takes place. The such condition leads to either the increased production of the uric acid or else a decreased clearance of the uric acid. So if it is either one of them, then it leads to the secondary gout. But if it is both, then it leads to the primary gout. The hereditary disorders associated with gout takes place in the three state. One, it results in the HTPRT deficiency, that is salvage pathway of the purine metabolism, as well as the superactivity of the PRPP synthase, and finally the glucose 6-phosphate deficiency that usually people seen it in the von Gus disease or the glycogen storage disease type 1. And there is one more type of a gout called a familial juvenile gout or the familial juvenile hyperuricemic nephropathy. This is an autosomal dominant disorder, and this occurs due to the severe renal hyperexcretion of the uric acid. And this presentation usually occurs at the puberty or to the third decade, and also been reported in infancy. That is uricosauria, and then hyperuricemia is seen, and then the gout is also seen, and then the familial renal disease and low urate clearance related to the GFR, and then the renal failure also people have seen it in the adulthood. The common treatment, if you see in the case of the gout, people usually provide the allopurinol. The reason is allopurinol is analog of this hypoxanthine. So whenever this allopurinol is given in the body, it is a strong inhibitor of the xanthine oxidase, and the xanthine and then the hypoxanthine, which are soluble, which will be accumulated in the body, and it cannot be converted to the uric acid, and finally which are accumulated and then they excreted. The reason is these are soluble, whereas the uric acid is insoluble. The common treatment includes the uh, people usually give the allopurinol, and then sometimes they give the other kind of drugs like the NSAIDs drugs that will reduce the pain, such as the probenecid and then benzbromarone and then RDEA 594. And then sometimes people usually give the xanthine oxidase inhibitors to reduce the production, and in uric acid to facilitate the excretion by the conversion to allantonin. And finally, people usually do the surgical removal of the tophi, that is a urate stone. So this, you can see people have removed it using the surgical procedure. And there are inborn errors related to this uric acid metabolism. The first one we have seen that, which is a superactivity of PRPP synthetase. So the PRPP synthetase that we have seen it in the first stage of the purine metabolism, where this ribose 5-phosphate plus ATP that will be converted to the PRPP, that is a phosphoribosyl pyrophosphatase. So what will happen here? Whenever the PRPP is synthesized more, then the purine metabolism will be increased. The increased purine metabolism leads to the increased catabolism that finally leads to the uric acid production. And then the second one is the adenosine lyase deficiency, where people usually seen this adenosine lyase that will synthesize the adenosine to the adenosine monophosphate. In general, affected individuals may have the mix of the neurological symptoms, which usually includes the abnormalities with the cognition, movement, and then artistic features, epilepsy, muscle wasting, and then the feeding problems will be then in the adenosine lyase deficiency. There are some immunodeficiency disorders related to the purine metabolism. The one such example is ADI deficiency, that we all know that it causes the SCID, that severe combined immuno deficiency. So what it does, whenever the adenosine cannot be converted to the inosine, this leads to the inhibition of uh, ribonucleotide reductase that in case are inhibiting the DNA synthesis. And finally, therefore, the WBCs cannot proliferate, such as the T cell and then the B cells functions are defective. So that leads to the case called severe combined immunodeficiency. So the number of T cells or the B cells are not there in the body. That leads to the other opportunistic disease like pneumonia, chronic diarrhea will take place with impeded growth and compared to the healthy individuals. And additionally, neurological problems also takes place, such as the developmental delay, motor function disorders, and then the hearing loss often occurs. This SAD is not treated in patients or in children. That leads to the death, usually the patients survive only the year or two without any kind of medications. And there is one more enzyme deficiency, that is a purine nucleoside phosphorylase deficiency. This also almost similar to the SCID, but only the T lymphocyte function is affected, not B lymphocyte things are affected. So, which is a milder version of the, or it is not so severe when compared with the SCID. And one more enzyme that we talk, that is a myoadenylate deaminase deficiency, that leads to the muscle adenosine monophosphate deaminase deficiency. This genetic disorder caused by the mutation in the gene AMPD1. So that interface with the muscle cells processing of the ATP, the key energy molecule of a cell. So due to the lack of this enzyme that converts the ATP to the inosine, and then the ammonia may present no symptoms, or it may cause the excess inducing cramping in the patients. So if you see AMP deaminase, what it will does, this AMP will be converted to the NH3 as well as the ionic acid. So the disorder is diagnosed through the accumulation of the ammonia or inosine monophosphate in the muscle tissue, and treatment typically consists of the exercise and then the modulation. And there is a final disorder in the purine metabolism, that is xanthine oxidase deficiency, otherwise the xanthinuria. This is a rare disorder that people have seen where the xanthine levels are upregulated. This leads to the more levels of xanthine and then the hypoxanthine in the body, and that leads to the xanthine hypoxanthine stones. And the blood uric acid level is very very low. There is high level of urinary excretion of xanthine. There is a reduced excretion of the urinary uric acid. Additionally, this condition can lead to the muscle disorders due to the deposit of the xanthine in the muscle. And then the treatment of the condition involve the avoiding of foods and then drinks that contains the xanthine derivatives such as coffee, tea, and then colas, and then maintain the high fluid diet. And there are certain disorders related to the salvage pathway where this hypoxanthine guanine phosphorylic transferase, that is SGPRT, is deficient in the body. That leads to the Lesch-Nyhan syndrome or the partial deficiency. And there is one more deficiency called adenine and phosphoryl transferase deficiency, that is APRT deficiency. This leads to the accumulation of the insoluble purine, that is 2,8 dihydroxyadenine. It can result in the nephrolithiasis, kidney stones, and then acute renal failure. So one such a disease that we see that is Lesch-Nyhan syndrome, that caused by the deficiency of an enzyme that is called HPRT, produced by the mutation in the gene located on the X chromosome, and this is otherwise called as a X-linked recessive disorder, which leads to the hyperuricemia and then the hyperuricosuriasia. And symptoms often include the severe gout, impaired muscle control, moderate mental retardation, and kidney problems. And these complications frequently emerge in the first year of life. Neurological symptoms can include the facial grimacing and then the involuntary breathing, aggressive behavior, self mutilation, and then the repetitive movements people have seen. And the mental deflects and the behavior do not react well to therapy. There is no known cure so far in the Lesch-Nyhan syndrome, but many patients can survive to adulthood. And we have seen that how this one gets disease or is glucose 6 phosphatase deficiency leads to the gout. So whenever the glucose 6 phosphatase deficiency is there, this glucose 6 phosphate will enter the HFP shunt pathway. That leads to the high ribose 5 phosphate synthesis, then the increased PRPP synthesis, then the purine synthesis will be increased, then finally uric acid synthesis will be there. And in addition to that, this glucose 6 phosphate will be converted to the lactic acid, which competes with the uric acid for the excretion, resulting in the uric acid retention in the body. There is one more disorder called hypouricemia that leads to the decreased uric acid formation and then the excretion in the body. The serum uric acid level, if you see, which is less than 2 mg per dl. So this gene responsible for this kind of disorder is called URAT1. And there are some drugs also contribute to this kind of deficiency, that is uricosuric drugs are the common in vegetarians due to the low purine content of the most vegetarian diets, and it has no recognizable symptoms that require the treatment. The next topic that we see, that pyrimidine metabolism. The pyrimidines are cytosine, uracil, as well as the thymine in the body. The raw material needed for the pyrimidine synthesis are three. One is glutamine, carbon dioxide, and then the aspartic acid, which requires the ATP for its synthesis. and which is quite different from the previous metabolism that we have seen. That is, ribose is needed in the case of the purine synthesis, whereas in the pyrimidine synthesis, which is not required as a PRPP. This whatever the pyrimidine that we are talking about, it will be synthesized as the UMP, that is uridine 5 prime phosphate, which is a precursor of the other pyrimidine nucleotides. The de novo pathway of pyrimidine synthesis requires the six different steps or six unique steps in the prokaryotes, whereas that is controlled with the three steps in the eukaryotes. That we will see it in the upcoming slides how it is happening. The first step is where the glutamine as well as the bicarbonate ions are combined using the carbonyl phosphate synthetase. Two enzyme is present here, which is really important for the step. So that requires the ATP and finally converts the glutamine to glutamate, and then the bicarbonate which binds with this amine group, then finally it forms as carbamyl phosphate. This carbamyl phosphate, in the next step, where in the presence of aspartate transcarbamylase, it will be converted to carbamyl aspartate. So from the carbamyl aspartate, from the step 2, it will be converted to L dihydrourate in the presence of dihydrourate enzyme, where one molecule of H2O will be removed from the system. The next case, this L dihydrourate in the presence of dihydrourate dehydrogenase, then finally the uric acid will be formed in the fourth step. From the fourth step, uric acid will be converted to the uretidine 5' monophosphate in the presence of uratate phosphoribosyl transferase. Here, this phosphoribosyl 5' phosphate will come. This will be converted as PPI, and then finally the inorganic phosphate will be synthesized. And then OMP will be synthesized. From the fifth step, this OMP will be converted to the UMP, where one decarboxylase enzyme takes place. That decarboxylase complex. Then finally it forms uridine 5' monophosphate. So this happens in the prokaryotes, whereas in the eukaryotes, the first three steps, that whatever we have talked about, the carbamyl phosphate synthetase, aspartate transcarbamylase, and then the dihydrouretase, exist at the dihydrouretase synthase multifunctional enzyme, whereas the 5 and 5, 6 steps are catalyzed by the bifunctional enzyme called UMP synthase. So once the UMP is formed in the system, it will be converted to uridine diphosphate, which is none other than the UDP, and then it will be catalyzed by the enzyme called UMP kinase. This happens in the presence of ATP. And again, uridine diphosphate will be converted to uridine triphosphate by the enzyme called nucleoside diphosphate kinase. And finally, uridine triphosphate will be converted to cytidine triphosphate, and in the presence of CTP synthetase, where methyl group donor will be the amine group donor will be the glutamine. Here also one ATP molecule will be used. So if you see the conversion of the UTP to the CTP by CTP synthetase, here this oxygen molecule, whatever it is there, that will be converted to the amino molecule in the presence of CTP synthetase enzyme. And finally, here so far we have seen the ribonucleotidase and how it will be converted to the deoxyribonucleotidase. It's a different story. So where the 2' will not have the oxygen, then that's why it is called as the 2' deoxyribonucleoside. So here the 2' is whatever it is there. The oxygen molecule will be removed. That takes place by the enzyme called ribonucleotide diphosphatase. So how it happens? This ribonucleotide reductase enzyme will be there, that will reduce the ribonucleotide diphosphate into the deoxyribonucleoside. How it happens exactly? Whenever any kind of this ribonucleotide reductase is there, which is in the reduced form, that will be converted to the oxidized form as an inactive. So this H2 molecule and then this O molecule will be combined and then excreted as H2O. This oxidized form should be reversed back to the reduced form to the to make more deoxyribonucleotide. That will be done by the thioredoxin. So the thioredoxin is in the reduced form will be converted to the oxidized form to give two H2 molecules to the enzyme. And this oxidized form again converted to the reduced form of thioredoxin that will be run by the thioredoxin reductase that happens in the presence of NADPH as well as in the FAD. So it's a series of reaction which will take place then finally forms the deoxyribonucleosides from the ribonucleotides. And we have seen the carbonyl phosphate synthetases in the system, and it has the two different kind of enzymes that we have seen. That is CPS1, which is in the urea cycle, and then second one is the CPS2. The CPS1 usually uses ammonia bicarbonate, ammonia as a nitrogen source to synthesize the carbonyl phosphate, whereas this CPS2 usually uses the glutamine as a nitrogen donor to form the carbonyl phosphate in this case. So the regulation of the pyrimidine biosynthesis, if you see, that is a CPS2, which is allosterically regulated by the PRPP, and then the IMPR activators, and several pyrimidines are inhibitors, that is UTP and then the CTP. And there is one more enzyme called aspartate transcarbamylase, which is an important and then regulatory point in the prokaryotes that catalyze the first committed pathway step. And then the allosteric regulators like phosphoregulators are ATP, and then the negative regulators are UTP and then the CTP. So this is the regulation of the pyrimidine nucleotide biosynthesis in E. coli, where this UTP will be the negative regulator of ATPase, whereas in the CTP also do the same thing. But the CTP synthase is getting activated by the GTP, and then the ATP, this aspartate transferase also will have the ATP as a stimulator, and CTP itself is an inhibitor of the CTP synthetase enzyme. And if you see overall, the purine synthesis requires the synthesized on the PRPP, but pyrimidine first synthesized, then it is added to the PRPP, and then regulated by the GTP and then the ATP. But pyrimidine biosynthesis are regulated by the UTP and generates the IMP, whereas in the generates the UMP as well as CMP, and this requires the energy, and both requires the energy in the form of ATP. So these are the regulations of the deoxynucleotide formation, whereas the CDP to the DCDP, and then the UDP to the DUDP requires the positive regulator of ATP and then the ATP. But inhibitor is DATP, DGTP, and DTTP, and here it is DATP and DGTP. And say ADP will be converted to DATP in the presence of the positive regulator DGTP. But whenever the inhibitor present, that is DATP and then ATP, this will not be occur. In the GTP case, which will be converted to the DGDP, but positive regulator is TTP, but the negative regulator is DATP. So the pyrimidine catabolism takes place, which usually hydrolyzed to nucleosides as well as the inorganic phosphate. Then it will be converted to the thymine as well as the uracil or the deoxyribose 1-phosphate are produced. Finally, the catabolism of the thymine and the uracil bases ends with the intermediates of the central metabolism called carbohydrate metabolism. How usually takes place? This uracil and then the thymine in the presence of dihydrouracil dehydrogenase will be converted to dihydrouracil or the dihydrothymine. And in the presence of dihydropyrimidinase, it will be converted to uriidopropionate or uriidioisobutyrate, and uriidiopropanase will come and it will be converted to the beta alanine and then beta amino isobutyrate. Finally, it will be converted to acetyl CoA and then the succinyl CoA, which will enter the TCA cycle or the fatty acid metabolism or carbohydrate metabolism at the end of the day. And one such disease related to the pyrimidine metabolism is uric acid urea. That usually happens in the absence of OPRTase or OMP decarboxylase or UMP synthase, which is inherited as autosomal recessive disorder. The clinical features related to this disorders are retarded growth, severe megaloblastic anemia, crystals excreted in urine, crystalluria may cause the UTI, and then the treatment usually the administration of the uridine monophosphate or uridine triacetate. How this happens whenever the UMP synthetase is not there, then it leads to the uric acid urea. So if you give more number of UMP or uridine acetate, it will be do the feedback inhibition. So that will not form the carbonyl phosphate. Then finally uric acid will not be formed. Then the uric acid formation, if it is not there, then there is no need of UMP synthetase. So that's why this UMP is given as a drug for uric aciduria. So there are different uric aciduria, type 1, type 2, and then the uric acid urea cycle defect or the beta amino isobutyric aciduria. You can go back to the slides and then check what are the different disorders are related to the pyrimidine metabolism. Next, we are going to see inborn errors of metabolism that usually underrated case of the metabolic disorders leads to the permanent mental retardation. In most of the cases, inborn errors of metabolism leads to the mental retardation. The first scientist who understood this kind of inborn errors of metabolism is by Sir Edward Garrett. So he gave an explanation for 3 or 4 disorders that is otherwise called as the Gerard's tetrad, that is alkaptonuria, albinism, and then the pentosuria, and then the cystinuria in 1909. So the importance of the early detection, if you see, there are certain inborn errors of metabolism which can be treatable in the case of early detection. Suppose if it is in the late case, then it causes the irreversible clinic effects. So the specific diagnosis may either to stop more negative consequence from the occurring in the some diseases or advise against the triggering factor. For example, acute porphyriasis, cystinuria, hemochromatosis, and then the glucose phosphate dehydrogenase deficiency are few examples. So when we have to consider this a metabolic disorder of anything, there should be a serious neonatal presentation and biochemical abnormalities. Liver disease or the dysfunction should be there in the case of the metabolic disorder, or neurological features, and then the cardiac features, skin and then eye symptoms should be there. Chorus facias will be there, and then the signs of the storage disease should be there in the case of the metabolic disorder. Often these symptoms are occur in the child stage. Otherwise, the child should be healthy. So there are different metabolism that should be considered as the inborn errors of metabolism in the body. The list of metabolism that we have metabolism as well as the inborn errors of diseases that we have given it in the tableted form. These are the amino acid ureas, and then these are the organic acid ureas, and then the glycogen storage diseases, and then the sphingolipidosis. This you can go back and then see, and then read the slides. What are the different disorders are there in the different metabolism, and finally the mitochondrial syndromes presenting in the child to adult. Then finally it ends with the mucopolysaccharide doses. How to what to say diagnosis these kind of inborn errors of metabolism? There are certain metabolism which will have certain urine order. For example, the glutaric acidemia, which has the sweetie feet, and then the maple syrup urine disease will have the maple syrup color, and then the hypermethanomia will has the boiled garbage smell, and phenylketonuria will have the mousy or musty urine odor, and trimethylaminuria will have the rotten fish disease kind of smell it comes. So other symptoms, if you see how to study these kind of disorders, that can be the plasma ammonia we can see in the case of the organic aciduria, and then the urea cycle disorders, and then the plasma lactate or the pyruvate in the case of the lactic acidosis, and we can check the 3-hydroxybutyrate, and then the free fatty acids we can see, and then the quantitative or the semi-quantitative analysis of the plasma, and then the urine amino acids that we can see, and finally the urinary or the plasma organic acid analysis we can go, or the urinary mucopolysaccharides, and then the oligosaccharide screening test or the galactosemia screening test we can go. And usually people will go for the genetic counseling to stop this kind of inborn errors of metabolism. This given to the patients or the couples who are advanced maternal age at more than the 35 years of age, positive maternal serum screening, and then the patient or the family member with a known Mendelian disorder or the prior pregnancy with a chromosomal disorder, and then family history of the mental retardation or the birth defect, and fetal anomalies by the sonogram, and then recurrent pregnancies or the loss or still birth, infertility, ethnic based risk carrier screening, and the consanguinity will be there. How usually people will go for the screening? Then the first diagnosis will be the prenatal diagnosis, where people will take the amniotic fluid from the trophoblast, then it will be sent for the gene deficiency test. And once the baby is born, and then after the birth, people will usually see the electrolytes, glucose, ammonia, or as the lactate or the pyruvate ratio people would have seen in to detect the or to diagnose the inborn errors of metabolism. There is something called rapid urine screening test, which has the detection of the 35 inborn errors of metabolism. The list that we have given it here. If any kind of positive result is there, then that leads to the presence of inborn errors of metabolism in that patients. There are certain advanced levels of techniques are there, that is thin layer chromatography, HPLC for the detection of the amino acid ureas, GCMS for the detection of the organic acid ureas, and then the tandem mass spectrometry for the diagnosis of more than 45 inborn errors of metabolism using a single sample specimen. And there is one more technique called DNA hybridization or the probe analysis. This is very, very simple, which goes and binds to the DNA of interest. And if it is any kind of disorder is there, then it will give the color. So it is very specific and then the sensitive and then the prenatal diagnosis is possible and sample volume should be very, very less because it is based on the color or the sensor. And then the high infrastructure cost it has, because that's why we cannot go for the DNA hybridization of the probe analysis test and request the dedicated trained personnel and then expensive consumables. And we can go for the other kind of specific enzyme assays in leukocytes, plasma, or serum, or in the red cells. And if you see the therapeutic modalities, there are different set of therapeutic modalities will go. For example, the substrate deprivation will be there in the case of the phenylketonuria, galactosemia, maple syrup urine disease, or fructosuria. In the case of the cobalamin defects and the biotin defects, we usually give the high doses of the coenzymes because it cannot be synthesized in the body. And then the enzyme replacement therapy in the case of the Gaucher disease, and then the Fabry disease, and then Pompis, and then Harler's disease. And sometimes we will inhibit the substrate synthesis in the case of the glycosphingolipidosis or else the bone marrow transplants, or in the case of the liver transplant will go in the different disorders like Wilson's disease, alpha 1 antitrypsin deficiency, or the Kriegel-Nager syndrome. And finally, we can go for the gene therapy that will replace the wild type gene in the case of the mutated gene in the body. So this is still in the under the research stage, which is not approved by the any kind of government. Our next topic that we start with is the integration of the metabolism. So far we have seen all the metabolism, their different disorders, how it is related and everything. Now we are going to see the integration of the metabolism. There are different pathways that are really, really important in the body. In terms of the carbohydrate metabolism, the major pathways that we have seen gluconeogenesis, glycogenesis, glycogenolysis, and glycolysis, and minor pathways like HMP shunt pathway, uranic acid pathway, and then the lactose synthesis. In terms of the fat metabolism that we have seen, fatty acid synthesis, lipogenesis, lipolysis, fatty acid oxidation, ketogenesis, and then the ketolysis. And in terms of the protein metabolism that we have seen, the protein lysis, proteolysis, amino acid oxidation, urea synthesis, and then the protein synthesis that we have seen. There's something called integration of the metabolism at the cellular level. Usually, the conversion of the carbohydrates into fats happens, and then the fats into carbohydrates by the gluconeogenesis, and conversion of the carbohydrates into the proteins, and then the proteins into the carbohydrates. But whereas conversion of proteins to fats only takes place, fats to protein does not takes place. So that's why if you see, whenever the any carbohydrate comes in the body, it will be converted to the glucose, then finally it will go to the pyruvate. So pyruvate will be converted to the acetyl CoA, then it will enter the TCA cycle, and finally it will go for the energy production. Whereas the lipids, when whatever that we are taking, it will be converted to triacylglycerol, then the glycerol, and then the fatty acid will be converted by the breakdown, and finally the malonyl finally from the fatty acid it will form the acetyl CoA. This acetyl CoA, again it can form the malonyl CoA, then it can form the free fatty acids, then it can form the triacylglycerol. But in the case of the protein, protein will be converted to amino acid that can be converted to the pyruvate, acetyl CoA, or oxaloacetate. Then again it can form the protein. So this is why these different metabolism of protein, carbohydrate, and lipids are interrelated, and then there is an integration between their cellular level. The different organs that are involved in the body in the integration of the metabolism, such as the liver, brain, muscle, heart, and then the adipose tissue. One such example, if you see, The liver usually takes up the blood from the running system, as well as it is really really important for the glucose metabolism or any other metabolism per se. It usually delivers the glucose to the blood system, and then it takes the fatty acid, and then it gives the fatty acid, and it transfers the glycerol, and finally it produces the glucose, fatty acid, and then ketone bodies required for the heart. Then what are the adipose tissues? Then it will again produce the fatty acid. Then finally it gets into the heart. Then it requires for the energy requirement. And in terms of brain takes up the glucose from the liver and then the ketone bodies for its energy requirement. And finally, there is glucose alanine cycle will takes place where liver will give the glucose to the muscle and then muscle will again give the alanine to the liver. So these are the different role play takes place. We'll see one by one. The first organ that we are going to see is liver. So liver is the metabolic hub of the body. So it first access the to the ingested nutrients and then the supplies the fuel to the brain, muscle, and then the other organs. And this is a central role, plays a very central role in the regulation of the carbohydrate, lipid, and then the amino acid metabolism. So if you see the enzymes that has a high turnover when compared with the other enzymes. So one such example that we see the carbohydrate metabolism, where whenever the blood glucose levels are low, this liver glucose 6 phosphate will be converted to the blood glucose, then it will be given to the blood. Or else whenever the blood glucose is so high, then it will be converted to the blood glucose 6 phosphate, then it will go into the glycolysis and then the pyruvate if the energy requirement is needed. If the energy requirement is not needed, then it will be converted to the liver glycogen. The same glucose 6 phosphate will be converted to the will be directed to the pentose phosphate pathway to produce the NADPH or as the nucleic acid synthesis. And it forms the acetyl CoA as well, this glucose 6 phosphate. Then finally it forms the cholesterol, fatty acid, and then the triacyl glycerols. Whatever it is, this glucose 6 phosphate is really really crossroad for the any carbohydrate metabolism that really needs the efficient GLUT transporters called GLUT2, and it basically depends on the glucokinase enzyme in the body. The next metabolism is the fatty acid metabolism. So whenever the fatty acids are there in the liver, if it has to be given to the body, then it will be broken down or it will be combined to the plasma lipoproteins, and then the free fatty acids will be degraded. And if it is not required in the circulation, then it will undergo the beta oxidation. Finally, it forms the acetyl CoA, and then it can form the cholesterol and then the bile salts or the steroid hormones, or else it can go into the TCA cycle again, and then finally it will produce the ATP for the energy needs. And other metabolism like the amino acid metabolism, which can undergoes in terms of the production of the proteins, such as it helps in the production of the liver proteins, and it helps in the production of the nucleotides, hormones, and then the porphyrins. And it also helps in the plasma proteins and then the amino acids in the bloods, for example in the tissue proteins. Then whenever the amino acid is not required, then the deamination will take place. Then finally it forms ammonia. Then ammonia also will undergo the urea cycle. So this only takes place in the liver in the body. And finally the pyruvate will be formed. Then the pyruvate will undergo the acetyl CoA, then the TCA cycle, or in the case of glucose, and then the glycogen metabolism will take place. So these are the things all are integrated in the metabolism that we have been talking about so far. And we know that one such mechanism, this glucose metabolism as well as the glucose metabolism as well as amino acid metabolism are interrelated. From the liver, the glucose molecules are transported to the muscle, but whereas from the muscle, it takes through the glucose alanine cycle. So the alanine will be transported back to the liver. So that forms the pyruvate. That pyruvate in the through the gluconeogenesis, then it forms the glucose. So the turnover of the glucose will be same whenever this transport takes place. And if you see in the case of the skeletal muscle usually generates the ATPs as an immediate source of energy, and what's in the intermittent phase on demand. So there is a slow twitch muscle versus the fast twitch muscle. In the resting phase, if you see, the skeletal muscle uses the 30% of the oxygen consumed, whereas in the during the intense exercise, it uses maybe the 90% of the oxygen that we are taking. And then the resting muscle, if you see, that uses the fatty acids that as a major fuel source, and then ketone bodies, whereas the moderately active muscle consume the glucose and then the ketones as a fuel. If you see in the muscle fatigue situation, that usually begins about the 20 seconds after the maximum exertion, and the resting muscle contains about the 2% of glycogen, and then amount of phosphocreatine would be around 10 to 30 millimolar. Here, the glycogen usually sourced as a source of energy. During the intense exercise, the glucose 6 phosphate derived from the glycogen is converted to the lactate by the anaerobic glycolysis, and this requires the epinephrine stimulation. And if you see the heart muscles are quite different from the skeletal muscles that has to pump regularly. There should not be any kind of stop, and whatever the rhythmic condition is there, it should have the proper rhythmic condition. Otherwise, the heart would be stopped and the death occurs. So the cardiac muscle is the only muscle which uses the fuels mainly the fatty acids, but it also uses the glucose, lactase, and then ketone bodies as a fuel for this function. And in cardiac muscles, the mitochondria are much more densely packed in heart than in the other cells, and making up the nearly the half of the volume of a heart cell. So heart contains the negligibly energy reserves as a glycogen or lipid, although there is a small amount of the creatine phosphate, which is a high energy phosphate in the system. So the supply of the both oxygen and the fuels from the blood to the heart must be continuous to meet its energy demands. And the next one is the metabolism in the adipose tissue. The adipose tissue acts as a sensor for the glucose in the body. So whenever the glucose levels are adequate in the body, then whatever the glucose is there in the adipose tissue will be stored as a triacylglycerol. That we know in the previous lectures, whenever the triacylglycerol is not needed in the body, it will be converted to the fatty acids. Then this fatty acid will be travel to the liver. But whatever that takes place, only the 25 percentage will be excreted, whereas the 75 percentage of the triacylglycerol will go back to the adipose tissue. And there are two kinds of adipose tissue found in the body. One is the white adipose tissue as well as the brown adipose tissue. This brown adipose tissues are the thermogenic in the case. It has the protein called thermogenin. So usually any NADPH or FADH molecule will synthesize the ATP molecules in the body as a energy currency. But whereas in this thermogenin, which called as the uncouplers of the body, that will not allow the body to synthesize the ATP. Then the without ATP synthesis, the energy whatever it is there, that will be exhibited as heat. So that usually takes place in the cold temperatures, and then this occurs usually in the cold adaptation animals. And finally, in the brain is the place where high amount of glucose used in the body uh, in a day-to-day life. Usually, it takes around 120 grams of the glucose per day, and that consumes the equivalent to the 15% of the total energy consumed each day. And it is a highly aerobic organ, and then 20% of the total oxygen consumed by a human. And requirement for the glucose and then oxygen remains quite constant, and even at the rest or asleep. No significant glycogen or other fuel reserves people have seen, and supply of both oxygen as well as the glucose cannot be interrupted even for a short time. Otherwise, anoxic brain damage results. Brain can adapt during the fasting to use ketone bodies as a main fuel source instead of the glucose as a major fuel. Fats cannot serve as a fuel because blood-brain bacteria cannot pass this albumin access. And if you see the well-fed state, there are the different metabolic events takes place. One such example is insulin will be upregulated in the body. That leads to the glucose uptake by the muscle and then the adipose, both in the using the GLUT4 and then the GLUT2 transporters in the liver, and then the glycogen synthesis takes place, but the glycogen breakdown will be inhibited. Glycolysis takes place, fatty acid synthesis, and then the triacylglycerol synthesis takes place. Whereas, uh, if you are in the fasting state, then all those things, whatever we have said, that will be reversed. The glycogen breakdown will take place, glycogen synthesis will be inhibited, glycolysis will be inhibited, gluconeogenesis takes place, fatty acid mobilization will be takes place, and then the ketogenesis usually takes place. This is usually happens in the early fasting hours, that is 4 to 16 hours. After the fasting, that is usually overnight fasting, we will usually have the refed state, that is a breakfast time. So whenever usually you take the breakfast, the liver will not take the or absorb the glucose because that's still in the gluconeogenesis phase of 2 to 3 hours. And when liver has refilled the glycogen stores, blood glucose level still arises. Liver synthesizes the fatty acid from the excess glucose. And this is a different levels of metabolism of different biomolecules in the case of the starvation stage. So the glucose will be metabolized, or the plasma level of glucose you can see still after the 8 days of the starvation, but whereas the fatty acid levels are came down, and then the ketone body synthesis are upregulated after 8 days of the starvation or after the 3 days of the starvation, whereas the protein degradation takes place after the 2 to 3 weeks of the starvation, and the fatty acids whatever it is stored which will not be there, or the negligible levels will be there after the 8 weeks of starvation. So the starvation usually has the multiple stages. The starvation, which means the after the 1 or 2 days of the fasting. So the first one is the glycogenolysis takes place for the first 18 hours. Then the second stage will happen the gluconeogenesis. Once all the stored glycogen is depleted in the liver, then the gluconeogenesis will take place, and that also coupled with the glucose alanine cycle of the uh, muscle system. And the branched chain amino acids also liberated by the protein catabolism or utilized by the muscle to give energy. The third stage would be the lipolysis. Whatever in the skeletal muscle, heart, and then kidney will shut down their glucose utilization and will depend mainly on the fatty acid for the energy needs. Then ketone bodies provide the fuel for the tissues like heart muscles, skeletal muscle, and then to some extent the brain. And then the fourth stage will be the acidosis, where the ketone body synthesis is more. So the production of ketone bodies leads to the metabolic acidosis. The pH falls and then hyperventilation occurs as a compensatory mechanism. The fifth stage would be the death or the death stage, where the metabolic acidosis and dehydration, unless corrected efficiently, will lead to the death. A normal person has a fuel reserves to live up to 45 to 60 days. So these are the overall metabolism levels that takes place in the body. You can go back and then see what are the different metabolisms that takes place, how the low sugar or the how insulin and then the glucagon regulation takes place during the fasting as well as the well-fed state. So usually after the metabolic changes after the 3 days, the liver forms the high amount of this ketone bodies like acetoacetate and the beta hydroxybutyrate. So this synthesis is increases markedly, producing the state called ketosis. And gluconeogenesis depletes the supply of the oxaloacetate, which is essential for the entry of acetyl-CoA into the citric acid cycle. Brain begins to consume the significant amounts of the ketone bodies in the place of the glucose. A quarter of the energy needs of the brain are met by the ketone bodies. The heart also uses ketone bodies as a fuel. And after the several weeks of the starvation, the ketone bodies become the major fuel of the brain as well as in the other cases. Usually, the per day of the brain need would be the 120 gram of the blood glucose, whereas after the 2 to 3 weeks of starvation, it will become 40. The same goes for the muscle, so it will require only 20 gram, whereas in the well-fed state, it needs the 75 gram. So decrease in the nitrogen excretion occurs from the 20 to 15 gram per day to 3 to 4 gram per day, following to 4 to 6 weeks of starvation. So these are the different major fuels in the body. One thing I have to stress here: the ketone bodies will be the uh, energy fuel in the case of all tissues in the prolonged fasting state when compared with other things. So the same takes place in the cardiac muscle as well as in the skeletal muscle, that you can go back and then see what are the things are preferred fuel in the different fed state. So with that, we are in the end of the lecture. So we will start with the questions now. So which one of the following is our symptoms of gout? Are you there?
Yes.
Yeah.
Okay.
So
Yeah.
which one of the following is are symptoms of gout?
Symptom of gout. Um, which one of the following? Uric acid more than 8, joint
Hmm.
pain, monosodium.
No,
First three.
not a hypoglycemia.
No.
Yeah. Which of the following enzyme is inhibited in the well-fed state?
Inhibited. Glycogen phosphorylase. Yeah, glycogen phosphorylase and yeah, that one.
That only one?
Carnitine. Is it for the fatty acid synthesis one? Carnitine transferase.
Yeah,
Yes.
carnitine transferase from
Yeah,
the
fatty acid synthesis.
fatty acid degradation won't take place. Fatty acid synthesis takes place in the well-fed state. So
Yeah,
both will be
that one
inhibited.
will be there.
Uh,
Carnitine palmitoyl transfer is a different one. No, it's a.
yeah.
Hmm.
And which of the following is the cure for inborn errors of metabolism?